Hello, thank you for viewing my presentation and giving me the opportunity to share my work with you today. My name is McCain. I'm an associate professor in the Departments of Biomedical Engineering and Stem Cell Biology and Regenerative Medicine at USC. So my lab focuses on engineering organs on chips. These are biomimetic microscale models of healthy and diseased human tissues. We design these such that they can be modular, scalable, patient-specific, and provide quantitative readouts of tissue physiology. So we use a lot of different technologies in the lab to make these devices. So first, we try to implement patient-derived cells as much as possible. So for example, using induced pluripotent stem cells that came from patient skin fibroblasts and differentiating those into cardiomyocytes. We use a variety of biomaterials such as hydrogels where we can tune the elastic modulus to mimic healthy versus fibrotic tissue, for example. We combine those with different microfabrication technologies such as microcontact printing. So here you can see a square cardiac microtissue that we micropatterned on a soft polyacrylamide hydrogel. The cardiomyocytes adhere only to the micropatterned fibronectin, so we can make tissues with defined architecture and these kind of reproducible form factors. We also fabricate microfluidic devices in my lab for culturing either monolayers of cells or larger constructs such as explanted zebrafish hearts or organoids. We use micromilling as well as 3D printers to make these devices. And finally, we develop and implement different assays to quantify tissue physiology. So here you can see our muscular thin film assay to measure the contractile strength of an engineered muscle tissue, as well as assays to measure calcium signaling. So the applications for these technologies are as new model systems for elucidating mechanisms of human diseases to identify druggable targets, and to also serve as next generation platforms for personalized medicine and drug screening. So as a specific example, I wanted to share with you our work on engineering neuromuscular tissues on a chip. So our goal here is to engineer patient-specific neuromuscular tissues that can be stable in culture for multiple weeks so that we can get tissue maturation and also be able to identify structural and functional deficits that are relevant to diseases such as amyotrophic lateral sclerosis or ALS. So these diseases are really challenging to model in conventional model systems like mice because there's so much patient-specific variation. So there's a lot of different genes that have been associated with ALS and many mutations even within those genes. So it's not practical to be able to model them in mouse models. So we really need sophisticated in vitro models that can recreate neuromuscular tissues and allow us to interrogate the function and dysfunction in healthy and disease settings. So we've fabricated these micromolded gelatin hydrogels. So the hydrogel is soft and has an elastic modulus similar to muscle tissue. And we've micromolded it with these this grooved pattern that is about the width of a single muscle fiber. So when we culture muscle precursor cells on these, they fuse and align and form into elongated muscle fibers like you can see here. So these are actually engineered muscle that we got from chick cells, but you can see nice alignment of the fibers and relatively mature sarcomere structure. We also use our laser engraver to cut these cantilevers into the tissue, and then we can peel them from the surface and actually measure the contractile strength of the tissue and get a nice sensitive readout of tissue physiology. We've also co-cultured these engineered muscle tissues with human-induced pluripotent stem cell-derived motor neurons that we've gotten from our collaborator, Justin Achita, in the stem cell department here at USC. So you can see here examples of our tissues after 1 week in culture versus 3 weeks in culture. So the green are the motor neurons and the red show the muscle fibers. And you can see the motor neurons really integrating with the muscle fibers and ultimately forming these relatively mature synaptic structures that you can see in white here. We've also been able to evaluate synapse function using pouch clamp electrophysiology with our collaborator Dion Dickman in the neurobiology department also at USC. So here you can see recordings from the muscle showing the activity of the synapse, so signals coming from the motor neurons. And we've gotten relatively mature signals from these engineered tissues. So our next steps for this are to engineer tissues with cells from patients with diseases such as ALS to see if we can monitor the dysfunction that occurs based on patient mutation, and then also test therapies to see if we can rescue the dysfunction. We're also working on engineering three-dimensional tissues to improve tissue maturity. So I'd like to acknowledge our funding sources, especially the Eli and Edith Broad Foundation and the ALS Association that funded the work I just showed to you. We're excited to collaborate, especially with clinicians who have access to patient cells to build these disease models, as well as engineers that can add more sophisticated sensing capabilities into our devices. And we're also interested in partnering with pharmaceutical companies and other industrial partners to test and scale up our technologies. Select technologies in our lab are patented and available for licensing. So please contact me if you have any questions, and look forward to meeting some of you. Thank you.